In this video, we will continue our discussion about the contribution of ER stress and nociceptive neuron towards tumor-associated macrophages induced tumor suppression. In the previous video, we have ended our discussion where we have seen the results for the Western blot that shows that this TAM induced ER stress that finally triggers nociceptive neuron to trigger these immunosuppressions. So now they wanted to see what's the contribution of this ER stress and nociceptive neuron. So before going into that contribution state, they first validated the results, or they first validated these ER stress contributions through different approach. In the previous video, we have seen that they have tested the Western blot, but now they have tested the immunofluorescence in the TAM abundant case and the TAM poor case. So in the TAM abundant case, they are able to see the markers for the TAM seems like the CD68. So in the TAM abundant case, we can see the ATF4 signals are high, which is the red color, whereas in the case of TAM poor, where CD68 signs are not there, ATF4 signals are very minimum. And these things they have quantified in the patient's tissue. This is HNC, head and neck squamous cell carcinoma tissues. These are the results on the patient tissues. These things they have quantified, and they are able to see that the ATF4 signals are high in the TAM abundant case compared to the TAM poor case. Afterwards, they wanted to check the contributions of this ER stress and the nociceptive neuron in this tumor TAM induced tumor suppression. So they checked it from this mechanical sensitivity parameters, how much pressure this ER stress and nociceptive neuron contributes towards these tumors or on these tumors. So they checked the mechanical sensitivity, and they did the two different conditions. In first case, or in one conditions, they knocked down this nociceptive neuron using RTS based denervation, and here they killed the nociceptive neuron. And in the other case, they use the CRISPR-Cas9 strategy to knock down ATF4. And when they check the mechanical sensitivity, they are able to see that both in these SCC7 cells and B16 F10 cells, the mechanical sensitivity goes down in the ATF4 conditions compared to more significantly compared to the conditions where nociceptive neuron is killed using RTX. This indicates the contributions of the ER stress is more compared to the nociceptive neuron. Thereafter, they also checked the calcium influx, since this is also contributes towards, or this is also one of the signaling pathways that helps in this TAM induced tumor suppressions. And here also they are able to see that the experiment done is with conditioned media from the tumor associated macrophages treated with ATF4 knockout SCC7, and they did it also for the control where they now killed nociceptive neuron using RTX. And the results, they are able to see that the Capsaicin responsive neuron or calcium influx is significantly reduced in the ATF4 knockout conditions compared to the nociceptive neuron knockout conditions. So this in overall indicates that the contributions by the ER stress is significantly higher compared to the nociceptive neuron toward this TAM induced tumor suppressions. The calcium influx is also one of the indications that, because calcium influx usually goes up with ER stress, and since ATF4 is knocked down, the level of ER stress will not be enhanced, or will not be reached to an extent where it can be reached in the wild type conditions where the ATF4 is not knocked down. So this shows that this ER stress has a role, or this TAM induced ER stress within the tumors that contributes more towards this TAM induced suppressions. This is what is come out from these results. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. One thing I would like to repeat, that we are still discussing the figure 2, and we will see what will come out when we discuss the last sections of the figure 2. So far it looks like ER stress is triggered by the tumor associated macrophages. When the tumor associated macrophages are present around the tumors, that will cause this nociceptive neuron to trigger these tumor suppressions. However, the role by ER stress is significantly higher compared to the nociceptive neuron. This is what comes out till now. We'll discuss further results to understand what are the final conclusions from this figure 2 and also from this paper. So that's all for this video. Thank you for your time.